Hello, good evening, everyone. Very good evening to all of you, and warm welcome from Spark Academy. Hello, good evening, everyone. This is Vaishali Kamdar from Spark Academy, and yes, we'll so that we can start the class. Yes, can I have some quick comments if my voice is clear or my screen is visible? Any problem? Anything? Any problem is there, guys? Please inform me in chat section. Yes, can I have some quick comments, please? If you can see the screen and if you can hear my voice clearly. So yes, before starting the biological classification, the remaining portion of it, yes, we have completed Monera protista and Monera, see, all these things, two things we have completed. We are supposed to discuss today fungi and Plantae and Animalia and few more points. So we have discussed about the protozoans, different type of protists, and Monera also, and some history parts of the biological classification that also we have covered. We have discussed in the last class, right? So before starting the remaining portion of this chapter, let me tell you all of you that yes, you can follow us on sparkacademyonline.com, Instagram, Telegram, Facebook, any of the thing which you are using, you can follow us on everywhere. So you will see the tips and tricks we are recording reels and tricks and tips we are posting there. So you'll be getting all the type of notifications and all the type of updates everywhere. Okay, so this is our team. This is our team for EAMCET J and NEET. On daily basis, we are conducting live sessions. So right now, this time we have started Telugu sessions also. Monday, Tuesday is English, and Wednesday, Thursday is Telugu sessions. So in all these live classes, these are free live classes on daily basis. Okay, so for that you need to see. After that, you can subscribe to our channel, that is Spark Academy channel. This will be free live classes on YouTube. So on daily basis, we are conducting these classes in session. Doubt clarification is there. Free materials are available. Tips and tricks for EAMCET and J and NEET. Admissions are open right now for 2023 classroom as well as online teaching. So this is our schedule for YouTube sessions. So you can check out the schedule. So biology and math generally simultaneous 2 classes for MPC, BPC students separately. And Mondays, Tuesdays, English. So first biology session 5 to 6, then 6 to 7 physics, 7 to 8 chemistry. So this is me, Vaishali Kamdar, and this is my coupon code VAI10. So you can enroll Spark Academy right now. For that, you need to download the app from the App Store or Play Store, and you just need to focus on the logo first. So this is our logo. To look at the logo, to like the way we are teaching, you like the way I am teaching, then you can download this app and see the courses which are available right now. All the courses which are available right now, and then enroll. If you are enrolling and using this coupon code, that is VAI10, then you will be getting huge discount on all the courses available on that app. Okay, and you'll be enlisted in my student list. So if you want me to teach you offline also, and then join us right now. What are you all waiting for? So you can download the app and just check the courses available, long-term course and regular course, whatever you are looking for, whatever branches you are looking for, Medipatnam, Narayangura and ECL. So we have three branches, third branch is started just now only. So whatever is your convenience, you can join and enroll right now. So we have started long-term classes. So don't miss, if you don't want to miss the classes, right now only download the app and use this coupon code VAI10 and enroll right now. So yes, as we have discussed previously also, that biological classification, from that you can expect 3 to 4 questions. So around 12 to 16 marks. All these things we have discussed: kingdom Monera, kingdom Protista. Okay. Chrysophytes, dinoflagellates, euglenoids, slime molds, protozoan. This was done last class. Now we need to start from the kingdom fungi. You can pronounce it fungi, anything. Yes. Wait a second. So they are heterotrophic. They show us a heterotrophic mode of nutrition and saprophytic in nature. Saprophytic, sapros, that means rotten, and they are growing over the dead and decaying material. Most preferable habitat, that is dead and decaying material. That's why they are also known as saprophytic. So normally they will directly absorb the organic matter present in the thing which over they are growing. So that's why they are all known as saprophytic. They show great with diversity in morphology and habitat. Different type of morphology, you can starting from the unicellular to the multicellular filamentous growth they are showing. So you can observe them in different shapes, and their preferable condition, it is warm and humid place. Main preferable preference, it is humid place. Okay, right now fungal growth you can expect in this season. Right now it's rainy season, so you can expect more fungi to grow in this season. Okay, if you keep one roti outside in for 2 days, then you will find fungus growth over it, because right now it's a humid and warm environment. So in this environment, everywhere you can find the growth of the fungus. So there is no specific habitat for the fungus to grow. The thing they require, their preferable thing which they require, that is warmth and humidity. So even you can observe them growing over the shoes, growing over the onion, so bread, everywhere you can see the fungal growth. Even in gardens, you can observe growth of the mushrooms right now. So mushrooms are type of fungi only. So most of them show filamentous growth. Filamentous, that means they'll show hyphal type of tree-like structure. But remember that they are not autotroph. So that's why we have separated them from the algae, or you can say from other same type of filamentous growth type of showing algal. In Thallophyta, previously fungi were kept in Thallophyta only now, but they have a separate kingdom fungi, because they are not autotrophic. Never they have chlorophyll. They do not have chlorophyll at all. So non-chlorophyllous kingdom is kingdom fungi. Okay, most of them shows filamentous growth except yeast, Saccharomyces cerevisiae. So brewer's yeast, it is also known as baker's yeast. In bakery, in brewery, we are using Saccharomyces cerevisiae, that is yeast, which is showing, which is unicellular fungi. Okay, unicellular fungi. You can see it will look like this. This is budding yeast. This is how you will notice the budding yeast. Okay. So unicellular fungi not showing filamentous growth, but yes, due to this attachment of the cells to the mother cell, that is called how this budding type of structure is formed. When this type of budding type of structure is formed, you will see this type of hyphal type of thing, which is not actually proper hyphae. This is called pseudo hyphae. This is called pseudo hyphae, or you can say false hyphae. Pseudo, that means false, hyphae. Or pseudomycelium they'll show. So because this is not properly, see, it is just like that, that these cells remaining attached for the to the mother cell for some time, then they'll get separated. So they are not hyphae or mycelium. So false type of hyphae we can observe in when we see the structure, but actually that is not hyphal structure. They are unicellular. Then some hyphae are continuous tubes filled with the multinucleated cytoplasm. Some hyphae, they are continuous tubes. So that means there is no septum in it. For example, this is one hyphae. In that you will see a lot of nucleus. These all are nucleus. Lot of number of nucleus are there. So multinucleated hyphae. But there is no, this type of septum is not formed. The septum is absent. But this type of structure, it is known as multinucleated hyphal structure. It is known as pseudocytic hyphae. Okay, pseudocytic hyphae. Then walls of the fungi composed of the polysaccharide and chitin. Then reproduction. Fungi show the three different types of reproduction: vegetative, asexual, and sexual. Vegetative mainly by fragmentation, but yes, sometimes fission and budding also, like in case of yeast. You will observe budding also. Asexual reproduction mainly by spores called conidia or sporangiospores. Conidia, they are mostly exogenous, and sporangiospores produce inside the sporangia. So they are endogenous. Sexual spores, oospores, ascospores, basidiospores. Sexual cycle include three stages. These include three stages. Which three stages? Plasmogamy, karyogamy, and meiosis. Plasmogamy, that means fusion of only cytoplasm. For example, these are two cells. Okay, these are two cells of gametes, you can say. So cells or gametes, these will undergo fusion. Okay, so first only cytoplasm of these cells will fuse and nucleus will remain separated. So this stage, in this stage you can observe two nucleus in one cell. So that's why it is known as dikaryon or N plus N arrangement, N plus N stage. Dikaryon or N plus N stage, that is dikaryotic phase. Okay, after this karyon, that means nucleus will undergo fusion, lead to formation of zygote. So this is zygote formation. Okay, zygote will undergo next stage, that is meiosis. So first it is plasmogamy. Gamy, that means fusion of only cytoplasm, plasmogamy. Then nuclear fusion known as karyogamy. Okay, leading to next stage, it is meiosis. I hope it is clear. Yes, you can write anything in the chat section if you are there, anyone. I know many of you have joined. Is this concept is clear, everyone? Am I clear? Vegetative reproduction by fragmentation, fission, and budding. Asexual reproduction by conidia or sporangiospore. Sexual reproduction, sexual cycle, involves plasmogamy, karyogamy, and meiosis. Then phycomyces, first various classes of the fungi. In that first group, it is phycomyces. So first class, it is phycomyces. They are commonly known as algal fungi. Remember this name, algal fungi. So that's why phyco terms related to plants. So that's why they are also called algal fungi, or they are lower fungi. Or they are also known as lower fungi. Lower fungi or fungi. Yes, hello, Fedros, Mirza. Then mycelium is accepted and mycelium is accepted, not accepted, but yes, mycelium is accepted here. And xenocytic phase is there. That means what we have discussed, multinucleated stage. Septum is not there. Septum, that means partition. Partition is not there. Partition is not there. Septum is not there. That's why this type of multinucleated stage is known as xenocytic phase. Is that clear? Yes. For those it is clear. Then habitat, moist and damp place and decaying matter. So they will be growing on the moist and damp place and decaying matter. Asexual reproduction by zoospores. Always remember zoospores, they are motile spores. They will be having this type of flagella. Okay. Zoospores, they are motile. Aplanospores, they are not non-motile. Non-motile spores. Sexual reproduction. Sexual reproduction by zygospores. This will occur because of the fusion of the gametes. See, gametes will fuse to form zygote. You know that. But gametes, depending on the different shape and morphology and physiology of the gametes, there are three type of condition you can say fusion: isogamy, anisogamy, and oogamy. If both of them are morphologically same, gametes are morphologically same. Okay, morphologically same. Then that morphologically as well as physiologically also same. Both of them are motile, then both of them are motile. If only one is motile, it will be not like that. In anisogamy, they are morphologically similar but physiologically dissimilar. So you can see here similar gametes will fuse, then it is known as isogamy. Dissimilar gametes will fuse, then it is known as anisogamy. And if male gamete is like this, motile and small, female gamete is big and non-motile, this type of fusion is known as oogamy. This type of fusion is known as oogamy. So example of phycomycetes are mucor, rhizopus and albugo. Albugo is the parasitic fungi on the mustard leaves. So you can say yes, what we eat in winter, that is sarso da saag and makke di roti. So in that sarso leaves of the sarso, that is mustard plant. So sarso might get infected because of the albedo, and you can see fungal growth over the leaves of the sarso. So that is because of albedo. Mucor and rhizopus. Rhizopus is bread mold. It is also known as bread mold. It is growing over the bread. Then next it is ascomycetes. Ascomycetes, they are commonly known as sac fungi. They are unicellular or multicellular yeast. Multicellular can be penicillium, aspergillus, clavicus, neurospora, anything. Unicellular like yeast. Saprophytic in nature, parasitic decomposer, and also shows coprophilous nature. What is coprophilous? New term, guys. Coprophilous, that means growing on the dung of the animals. Why they are called sac fungi? Because during the sexual cycle, they will form this type of sac, and inside this sac, their spores are formed, and these spores are then dispersed when this sac will open. Okay, that's why they are commonly known as sac fungi in sexual reproduction. Asexually, they reproduce by conidia formation. What are conidia? It will look like this. For example, this is conidia. This is how exogenous spores are there. Exogenous spores are formed. This type of growth you can observe in case of Penicillium conidia. They reproduce through conidia. Okay, and this mycelium, this hyphae which bears this conidia, that is known as conidiophore. Conidiophore. So asexual reproduction by conidia produced exogenously on the conidiophore. Then sexual reproduction, sexual spores called ascospores. They are called ascospores, produced inside the endogenously. Look here how they are produced endogenously inside the asci, that is sac, which are arranged in the fruiting body. These sacs are arranged in a fruiting body, always fruiting body with the suffix carp. So these are known as ascocarp. Yeast, Penicillium, Aspergillus, Claviceps, Neurospora, morels and truffles, they are edible fungi. Morels and these all are ascomycetes, but morels and truffles, they are specially edible fungi. They are also known as delicacies. So this is about ascomycete, commonly called sac fungi. Next, it is about basidiomycetes. So basidiomycetes, mushrooms, bracket fungi, and puffballs. So mushrooms are species of agaricus. They look like mushroom and they are called mushrooms commonly. Bracket fungi, that means, for example, you see right now in this humid environment, this is the stem of the plant. Then you might see this type of bracket growing here. Okay, like this. So these are known as bracket fungi. Name of the bracket fungi is polyporus. Then puffballs, simply just like balls they are growing. Okay, white ball, you can see white shining smooth soft balls. They are known as puffballs. Lycoperidons, they are puffballs. Mushrooms, you all know about the mushroom, this type of structure, umbrella like structure of mushroom. This is known as basidiocarp. So commonly all these are basidiomycetes and commonly they are called as club fungi. Parasitic form is rust and smut. Rust, name of the rust is Puccinia and smut Stiligo. Mycelium is branched and septate. Here mycelium is branched and septum. Just like ascomycetes, it is not having this type of sinusoidal phase. It is branched with the single nucleus. Okay, branch is there and with single nucleus, that means septum is also there. Asexual spores are not found, but yes, fusion occurs between the somatic cells. Fusion occurs between the vegetative cells. So that's why it is also known as somatogamy. Whenever we use the term gamy, that means fusion. So leading to the dikaryotic stage, and then there will be formation of basidia. This basidia will arrange in form of fruiting body that is known as basidiocarp. Fruiting body that is known as basidiocarp. Okay. Now inside this basidia, there will be dikaryote stage here. Then fusion occurs in zygote formed inside the basidia. This zygote will undergo meiosis there only to form meiospores. It will form 4 basidiospores. Each zygote from that 4 basidiospores. You know that product of the meiosis, it will be 4 daughter cells only. Then next class, 4th class of the fungi, deuteriomycetes. They are also known as fungi imperfecti. Why they are called imperfect fungi? Why they are called imperfect fungi? Because perfect stage is missing here. Perfect stage is not there. What is perfect stage? That is sexual reproduction. So sexual stage is not there. Sexual reproduction is absent. You can see it is absent or present. Asexually reproduced by, they are asexually reproduced by conidia formation and exogenously. See, if this deuteriomycetes, they in their life cycle, somehow they will carry out sexual reproduction, then they will be placed in asco or basidiomycetes depending on what type of fruiting body they will form. Okay, so you can also say that they are ascomycetes or basidiomycetes only, but not showing sexual reproduction. That's why placed under this deuteriomycetes. Alternaria early blight, then Calectotrichum, red rot, and Trichoderma. These are an example of deuteriomycetes which does not show sexual reproduction in their life cycle. If they will show, then they will be placed in Asco or Basidiomycetes. I hope there is no doubt in these things. Now next kingdom is kingdom Plantae. So we will discuss this plant kingdom again in the next class. But yes, basic thing about the plant kingdom is the cell wall. Yes, plant cells, they are eukaryotic in nature. That means nucleus is bounded by the nuclear membrane and cell possess the cell membrane. Additional with the cell membrane, cell wall is also there to give rigidness to the cell. And the cell wall of the plant is made up of cellulose. Plants mostly show the autotrophic nature because of the chlorophyll present in them. They can carry out photosynthesis. Some plants are heterotrophic also, like parasitic plants, like insectivorous plants, in Venus flytrap and pitcher plant. All these are they are showing heterotrophic type of mode of nutrition. Main important characteristic of the plant is alternation of generation. Plant can switch their life cycle. Half stage of the plant kingdom Plantae, it is haploid and diploid. Haploid to diploid, diploid to haploid. They'll switch their life cycle. That is known as alternation of generation. Then kingdom animals, Animalia. So Animalia, they lack cell wall. We all lack cell wall. What type of storage form is observed in animals? We store our food in form of glycogen and fat. In animals, it's holozoic nature, holozoic mode of nutrition. That means we eat our food whole. We need to ingest our food and digestion occurs inside our body. Okay. Some animals, they shows locomotion also and well-developed neuromotor systems also developed, highly evolved animals. So without any exceptions, over all the animals, they are multicellular. So here we have completed 5 kingdom classification of Whitaker, and that Monera, Protista, fungi, Plantae and Animalia. So now few more points about this chapter. So that is virus. First it is virus. So viruses, they are not kept in the, they are, you can say, exception to the cell theory is virus. So they do not follow the cell theory. Okay, so you can see viruses actually there is controversies whether they are living or non-living, because inside the host they are living, outside the host they are non-living. So that's why viruses, the name virus comes from the meaning of it is venom or a poisonous fluid. This coined by virus name was coined by Pasteur. Then Mosaic disease recognized by Ivanovsky. Tobacco mosaic disease, it was recognized that it was, the causative agent is something else smaller than the bacteria. Why they have noticed that it is smaller than the bacteria? Because these things that was passing through the filter meant for the bacteria, to filter the bacteria. So they might be smaller than the pore size where bacteria cannot enter, but viruses can enter. That's why we can say that these are smaller in size. So mosaic disease was recognized by Ivanovsky. Then Bajering demonstrated that contagium vivum fluidum, that means contagious, infectious living fluid. If you extract the fluid of the infection, then this infectious fluid can cause the disease in the healthy animals or a healthy living thing. Okay, this was demonstrated by Bajering. Then Stanley showed that virus could be crystallized, and the crystallized thing was protein. So if you see the virus, they will show hexagonal type of structure, sometimes polyhedral structure, sometimes different helical structure. So mostly bacteriophage. That means virus which infect the bacteria. So if you want to see, the structure of it will be just like this. Hexagonal head with a tail pin, tail fibers, and inside that DNA or RNA as genetic material. So it is DNA or RNA. In viruses, they can have DNA or RNA as their genetic material, not both of them together. So this is how this virus will look like, and from using this tail fiber and tail pin, they'll attach to the bacteria. This is how they'll get attachment to the adherence to the bacteria, and then they'll enter their DNA inside the bacterium, and they'll use the machinery of the host cell to replicate their DNA. So they'll be not having their own replication machineries. No enzymes they'll be having. Only proteins and DNA. So that's why they are also known as nucleoprotein. Inside it is nucleic acid, outside it is protein. Right? Bacteriophage mostly shows they'll be having double-stranded DNA as their genetic material, but plant viruses will be having single-stranded RNA as their genetic material. In animals you can observe different type of genetic material. Some are having ssRNA, some are having double-stranded RNA, some are having double-stranded DNA also. Then viroids, these are only genetic materials without coat, without protein coat. See, this protein, this outside thing, it is known as, just a second. Outside thing, it is known as protein coat, which is made up of subunit called, that is also known as capsid. Protein coat, it is also known as capsid. And this capsid is made up of the subunit capsomer. So now there is no nucleic, you can say no protein coat outside, only nucleic acid is infectious, then it is known as virus. They are still smaller than the viruses because they'll be not having protein coat. So Danier discovered this viroids, only genetic material is infectious. Potato spindle tuber disease, in that this is the disease caused by the free RNA. So free RNA, free genetic material, it is viroids. Then prions, these are misfolded proteins. They become infectious because of the folding is not proper of the protein. You know that protein, see, after translation, whatever protein is synthesized, it is known as polypeptide chain. But then this polypeptide chain will undergo folding. First, it will form secondary structure, alpha helix and beta pleated sheet. Then tertiary structure, then quaternary structure. During this folding of the protein, there will be some error occurred, and because of misfolding, protein becomes infectious. So they are known as prions. Mad cow disease, scrappy disease in sheep, all these are prion mediated disease. Then last point of the chapter, lichens. Lichens will show, in lichens you can have, there are two components of the lichen. One is algae and fungi. So algae, that is also known as phycobiont, and fungi also known as mycobiont. So algae prepares, see, this is an example of mutualism. Algae prepares the food for the fungi. Fungi gives the shelter and absorb the water and nutrients for the algae. So that's why they live together in form of lichen. You can't differentiate which is algae and which is fungi. So commonly the unit of the algae and fungi together, or you can say mutualism between these two, they are known as lichens. So lichens, they are best pollution indicators in a particular area. If lichens cannot grow, then you can't grow the lichens, then your area is a very polluted area. So they are pollution indicators. So this is about this chapter, guys. If you have any doubt, you can ask me the question. I'm waiting for your doubts, if any, or you can just write in the chat that ma'am, no doubt. We can go ahead with the questions. Yes, those who have joined, you can say, you can write something in your chat section. Any doubt, anyone? Anything you want me to repeat or revise? I hope that everything from the concept part, it is clear. So now we are moving towards the question answer session. Easy peasy types of MCQs, PYQs I have included. So do not go anywhere. Do not go anywhere. Stay tuned here and don't go anywhere. We'll be solving the questions. So before solving the questions, moving towards the questions, yes, let me tell you all of you, yes, you can download the app Spark Academy and for the great discounts on our courses. So after that, you will get handwritten materials, study materials, PYQs, mock tests available. So admissions are open right now. You can join us. 3 branches, Narayangura, oldest one, and Narayangura, Mehdipatnam and ECL. So inter student, inter plus EAMCET, you can, those who are preparing for EAMCET JU and NEET, they can go for this long-term program 10 to 4. Regular coaching for those who are right now in 11th and 12th, they need to attend their colleges. So that's why evening timing for them 6 to 8 p.m. Then school tutorial. This is something new we are starting. We have started this year. School tutorial timing is 5 to 8 p.m. Link in the description box for the Spark store. So you can buy any of this thing from the Spark store. Long-term course, crash course, mock test, previous year solution, full-length test, study material also. So buy right now from the Spark store and don't forget to use the coupon code VAI10. You can see the coupon code here in the slide. And for online coaching is also going on daily basis. On the daily basis, I am conducting classes for biology, and we all are conducting all the classes for every subject. So yes, you can opt for online coaching if it is not feasible for you to come to the institute and attend the class offline. Then online is also a good option. You can go for it. If you are not in Hyderabad, if you are staying very far from the center, so you can go for this online coaching. So on daily basis, this is how we are recording the session, and this sessions will remain in the recorded form in the app. So you can check the recorded form of videos afterwards also. So these are live sessions. That means whenever we are conducting offline class or online class, so you will be having access to the, we are keeping it live also. So common live sessions, see this is how on daily basis we are giving assignments. Also we are checking that whether it is pending, it will show whether it is submitted or pending. Then exams, whether they have attempted or not, that will also show to us. And test result is conducted. This is how result thing you will get. Okay, time, how much time you have taken, total score, grade, everything. Why Spark only? Because we have highly experienced faculty, personalized coaching you will get, which you will never get in any other institutes. Process oriented coaching, personal mentoring, and the doubt clarification session also. Limited batch size, we have our method. Basic concept shortcut method. First we explain the basic concept, then we'll give the shortcut method to how you should memorize the things. Keys, how should prepare the keys. We are training our students that they need to prepare, they need to learn how to make the keys. And all model questions, exercise questions, PYQs, chapter-wise test, weekend test, grand test, test analysis, everything is done. Everything is done. Okay. Next, hybrid coaching. That means classroom plus online. When you are going for the hybrid coaching, you will be having access to the online classes also. So that means whenever you can't come offline, it's raining or something happened, and because of that, yes, you can join the online session also. So in hybrid, you will get more benefits. You will be getting books also, which in online you will get only online materials. Otherwise, it will be same for everyone. Doubt clarification, grand test, weekly test, everything will be same. Now it's about first question. So first question, pick up the wrong statement here. Wrong statement from this thing. This is not 2015 rupees. It is asked in the 2015. Pick up the wrong statement. Choose the wrong statement from here. Protista have photosynthetic heterotrophic mode of nutrition. Some fungi are edible. Nuclear membrane is present in Monera. Cell wall is absent in Animalia. So yes, I am giving you clue here to select the correct option. Yes, where is my cursor? Just a second. Yes, first question, answer number 1, please. Everyone. Wrong statement here. Protista have photosynthetic and heterotrophic mode of nutrition. Yes, that's right. Because chrysophytes and dinoflagellates, they are photosynthetic. Euglenoids, they'll show both of them. Heterotrophic, then they become saprophytic and heterotrophic. So this is correct. Then some fungi are edible. This wrong statement you need to choose here. Okay, some are edible fungi. Yes, agaricus or mushrooms. Molars and truffles, they are edible fungi. Nuclear membrane is present in Monera. This is wrong. That's why our correct option is 1C. Monera includes only prokaryotes. Prokaryotes. Prokaryotes, primitive type or primary type. Primary type of karyon. Karyon means nucleus. So no membrane bound nucleus in a bacterial cell. It will be like this. Freely floating genome in the cytoplasm. So that's why nuclear membrane is absent, not present. So C is the wrong statement. So our answer is 1C. Next, in which of the organism the cell wall forms the two thin overlapping shells which fits together? In which of the organism cell wall shows the soap box type of arrangement? Cell wall shows the soap box type of arrangement. That is fitting overlapping thing. Yes, correct answer. You're right. Correct answer here. It is chrysophytes. Chrysophytes, and that especially diatoms. Diatoms shows this type of overlapping shells type of cell wall. So answer is 2D. Then third one, choose the wrong statement here. Neurospora is used in the study of biochemical genetics. That's right. Morels and truffles, they are poisonous mushrooms. That is wrong. Morels and truffles, they are actually ascomycetes and they are not poisonous. They are edible fungi. So this is our correct option. Correct option is 3B. Then next question, which of the following are most suitable indicator for SO2 pollution in the environment? Sulfur dioxide pollution in the environment. So conifers, algae, fungi and lichen. So your answer is lichen, as the last point of the chapter is that these lichens can be used as pollution indicators. They will not grow in presence of more SO2. So answer is 4th D. Next question. Which one of the following statement is wrong for the viroids? So they lack protein coat. They are smaller than the viruses. They cause infections, and their RNA is high molecular weight. If you want to give it a try, guys, if you are answering, I'm waiting. So yes, correct answer. Right, correct answer is their RNA is high molecular weight. It is wrong answer. So we need to select the wrong one here. So correct answer is 5th D. Then next one, one of the major component of the cell wall of the most fungi is chitinous cell wall they'll be having. They have chitinous cell wall. So correct answer is 6A here. Next, which one of the following statement is wrong? Again, wrong statement. Cyanobacteria are also called blue-green algae. Cyanobacteria, they are also known as blue-green algae. That is right. Golden algae are also called desmids. That is also correct. Eubacteria are also called false bacteria. See, that is wrong. U means wherever we are using U term, that means true. True bacteria. So eubacteria are not called false bacteria, but they are actually true bacteria. Yes, Kiran. 7th question. C answer. Where were you, Kiran? So 7th question answer is 7th C. Now this is your homework question, so you can take the screenshot of it and answer. If you want to give me the answer, I am waiting for your answer. Chalo, which of the following components provide sticky character to the bacterial cell? Yes, I am waiting, guys. Right now only. We'll do homework in classwork form. Okay, I know this is homework, but as you haven't done the classwork yet, so you can do the homework right now. Okay. Okay, Kiran. Yes, I can see many of you have joined, but nobody is answering. So answer this question. Waiting for all of you to answer, guys. And see, whenever you are posting your chats, whenever you are writing anything in your chat section, then only I can see who has joined. Okay, otherwise I could not. I can see the numbers only, these many people have joined. So which of the following components provide sticky character to the bacterial cell? So nuclear membrane, plasma membrane, glycocalyx or cell wall? Yes, it is glycocalyx layer, or in that specially sticky characteristics, glycocalyx is mostly made up of the polysaccharide capsule and slime gives the stickiness to the bacterial cell. Correct answer is 1C. Next question. Viroids differ from the viruses having what? Next question. Come on, I'm waiting for your answer. DNA without protein coat. Viroid differ from the viruses. That means we need to think about the viroids. So A, B, C, D, they are characteristics of the viroids here. So DNA without protein coat, RNA molecules with protein coats, RNA molecules without protein coat, DNA molecules with protein coats. No, no, Kiran. See, we need to think about the viroids here. So viroids, they are only genetic material which becomes infectious. Genetic material which becomes infectious, and especially RNA. So in that, see, they'll be having RNA without protein coat. RNA molecule, they are actually RNA free RNAs which can be infectious. So RNA molecules, not this, with not this one, okay, RNA molecules without protein coats. So yes, you're right, Kiran, 2C is right. 2C is correct, not 2A. You were right. Next question. Which among the following are the smallest living things known without a definite cell wall, pathogenic to the plant as well as animals, and can survive without oxygen? So anaerobic, lacks cell wall, and smallest living cell. Correct, 3B is correct answer. Mycoplasmas, they are also known as pleuropneumonia-like organism. So mycoplasmas, 3B is correct answer. So your homework is done in classroom only. Okay. So do not forget to subscribe the channel and press the bell icon for the notification. Do not forget to do this, guys. Likes are very important. Don't forget to press the thumb icon for liking the video. Okay. If you like my way of teaching, if you think that your concepts are getting cleared by me, then please don't forget to press the thumb, or you can say don't forget to like the video. This is what our results, Spark Academy results, EAMCET results, Spark Academy results summary, highest rank, highest engineering and agriculture for EAMCET, JEE mains results, key numbers of the students studying in the Spark, average percentage of the Spark students 87 percentage, pass percentage 98 percentage, rankers toppers 7 to 8, average percentage. You can be this part of this. You can be increase this number of the toppers. So pass percentage of the TS students 67 percentage. So this is the detailed result of them. Detailed result of the NEET 2022. JEE EAMCET NEET admissions are open right now. 3 branches and online sessions are also there. Classroom and online coaching is going on. For regular admissions are open right now, so you can call on the number flashing in your screen. After school tuitions also we have started. Thank you so much, guys. Do not forget to like, share and subscribe, and do not forget to write the comments. Thank you so much.